Hi, this is Maggie Byers, and this is my Exam 9, Question 1. So, the immune system operates on a integrated and highly coordinated defense network. So, it's designed to protect the body from a wide range of pathogens, and it consists of two main components. So, it consists of the innate immune system and the adaptive immune system. The innate immune system acts as the first line of defense, and it responds within minutes of infection. So this includes the physical barriers of the skin, like the skin, and chemical barriers like the antimicrobial peptides and cellular defenders like neutrophils, macrophages, and natural killer cells. And these elements recognize broad patterns found on the pathogens called PAMPs, which is pathogen-associated molecular patterns. And these work to immediately to contain the infection. And then the adaptive system, this takes more time to activate because it depends on the recognition of specific antigens. And then once it's activated, T cells and B cells can generate a targeted response to that exact invader and create memory cells that make future responses faster and effective. So what's really important in these two systems is that they don't work in isolation. They're in constant communication. So the innate immune system provides signals and antigen presentation that is necessary to activate the adaptive response through cytokines and antigen presenting cells like dendritic cells and macrophages. And then the adaptive immune system enhances innate defenses by producing cytokines that increase activity of macrophages and antibodies that mark pathogens for destruction. So they aren't just one thing, they work together to constantly build each other up and form immunological memory that protects us from reinfection. So I'm going to talk about the first line of defense, and I meant to put physical barriers. So the first line of defense is like the skin and the mucous membranes. These form together the first line of defense in the innate immune system, and these barriers are always active. They're non-specific, meaning they protect a wide range of potential pathogens without needing prior exposure. So the skin's outer layer is the epidermis, and it is composed of keratinized cells. These act as a physical shield, which makes it difficult for microbes to penetrate. And then the mucous membranes, these line the respiratory and digestive and urogenital tracts. These secrete mucus and sticky, which is a sticky substance that traps pathogens and debris. And then we also have cilia and the respiratory tract. These move this mucus upward and outward, and this helps clear microbes from the airways. And in addition to this, we have secretions like tears, sweat, saliva. These contain enzymes like lysozyme, which breaks down peptidoglycan walls, causing bacterial lysis. So these all make up the first line of defense. And these are features of the innate immune system, like I talked about. But they also play an important role in supporting the adaptive immune system. So when these pathogens manage to breach these outer defenses through cuts or inhalation or ingestion, they encounter innate immune cells like macrophages, dendritic cells, which recognize and engulf the invaders. And this interaction bridges the two systems. So innate defenses contain and limit infection early on, but adaptive immunity develops a specific targeted response and immunological memory for future protection. So I'm going to go a little bit more in about the chemical and physiological barriers. So beyond those physical barriers like the skin and mucous membranes, the innate immune system relies heavily on chemical defenses. So these chemicals act as the body's biochemical shield. So we talked about lysozyme, which is an enzyme found in tears, saliva, mucus. It breaks down the walls and causes bacterial lysis. And then we also have urine, which helps flush the pathogens out of the body. Sebum, which is used, it has from skin glands. It creates an acidic environment and flushes away. It goes with sweat. This is on the skin surface. It inhibits microbial growth. And the stomach also has a low pH and digestive enzymes. These destroy ingested pathogens. And we've already talked about pretty much all the other chemical responses. So I'm going to talk about the inflammatory response. So, inflammation is one of the most critical and immediate innate immune responses to injury or infection. So, when tissue is damaged, cells will release chemical signals called inflammatory mediators, and these include histamine, bradykinin, and prostaglandins, and these increase blood vessel permeability and vasodilation. This causes more blood to flow to the affected area, which is why we see redness and warmth at the site of inflammation. And this increased permeability allows plasma proteins and immune cells to exit the bloodstream, and neutrophils are the first responders moving through the vessel walls by diapedesis to engulf pathogens or debris. So soon after macrophages arrive to continue phagocytosis and release cytokines that attract more immune cells, and this ensures a more coordinated and localized response. And inflammation is primarily an innate defense mechanism, but it plays a crucial role in activating the adaptive immune system. So APCs that we've talked about before, these arrive during inflammation, and these include macrophages, dendritic cells. They process and present pathogen antigens to T lymphocytes, and these mark the beginning of the adaptive immune response like we talked about earlier. So cytokines are released during inflammation, and they stimulate proliferation of helper T cells and B cells. So this connection ensures that once the innate system identifies and contains the invader, the adaptive system is primed and to develop pathogen-specific immunity and immunological memory. So I'm going to talk a little bit more about fever and microbial proteins. Fever, we talked about, is one of the most effective mechanisms in the innate immune system. And when pathogens are detected, immune cells like macrophages, neutrophils will release interleukin 1 and tumor necrosis factors. So these pyrogens signal the hypothalamus to raise the body's temperature to a set point, and the higher the temperature, the harder it is for microbes to replicate efficiently. And this also enhances the activity of white blood cells. So especially this includes phagocytes and lymphocytes, and fever also promotes the production of interferons, which are proteins that inhibit viral replication. So while uncomfortable fever is actually a critical part of the innate immune systems to attempt to create an environment less favorable to pathogens, but more favorable for immune efficiency. So now I'm going to touch on phagocytosis. So phagocytosis, this is the process when the body engulfs and destroys and invades microbes, and the pathogens enter. When the pathogens enter, neutrophils are the first responders to the infection site, and they use chemotaxis to follow chemical signals that are released by damaged cells and other immune cells. So once they reach the target, the neutrophils will engulf the bacteria and into vesicles, which are called phagosomes. So these infuse with a lysosome to form a phagolysosome, where the pathogen will be broken down, neutralized, and then die off. So now we're going to talk about natural killer cells. So these play a key role in early viral defense. So these are unique components of the innate immune system because they perform surveillance duties that are similar to cytotoxic T cells in the adaptive immune system. So they will continuously patrol the body and scan for host cells that show signs of stress, infection, or transformation, like those infected with a virus or a cell that's becoming cancerous. So unlike T cells, the natural killer cells don't require a specific exposure to a specific antigen to act. Instead, they rely on recognizing the lack of normal surface proteins such as the MHC1 class 1 molecule or the presence of an abnormal one. So when natural killer cells detect these changes, they can release cytotoxic granules containing perforin and granzymes. These will penetrate the target's cell membrane and trigger apoptosis. So while they belong to the innate immune system, natural killer cells function an important bridge to the adaptive immunity, and the early response helps contain infection during critical period before T and B cells are activated. So In addition, natural killer cells, they also secrete cytokines, which act as, or such as interferon gamma, which help enhances macrophage activity and promotes the activation of cytotoxic T cells later on. So this cooperation ensures that by the time adaptive immune responses develop, the pathogen spread is limited and antigen presentation is already underway. So now I'm going to talk about the transition to adaptive immunity. We've talked about innate immunity and how it connects to adaptive immunity, and now I'm going to go more in depth with adaptive immunity. So the innate immune system serves as the body's first responder, so it reacts within minutes or hours of infection, and it uses general defense mechanisms like inflammation, phagocytosis, and the release of antimicrobial proteins to contain pathogens. But a microbe is especially virulent or numerous. If they are, the innate immune system will not be enough alone. So when this happens, it sends chemical signals like cytokines or interleukins to recruit and activate the adaptive immune system. This is the next level of defense, and it takes longer to mobilize because it relies on the activation and selection and proliferation of specific T and B lymphocytes. So once it's activated, the adaptive immune system will respond precisely to that microbe and ensure a highly targeted and effective attack rather than a broad, non-specific one. So this transition from innate and adaptive immunity represents the two branches. So the innate system acts as the initiator and identifies and presents antigens through APCs, effectively teaching the adaptive system what to target. And then the adaptive immune system then refines the response using cytotoxic T cells to destroy infected host cells and plasma cells to secrete antibodies that will neutralize pathogens. And this is advantage to the system is memory cells because they remain long after the infection is gone and ensure that if the same pathogen returns, the body will create a faster and stronger response before the symptoms can even appear. So now I'm going to talk about antigens. So antigens are molecular identifiers. They are unique and they're found on the surfaces of bacteria, and they act as like the fingerprints to distinguish self from non-self. And when a pathogen invades an antigen-presenting cell, like macrophages and dendritic cells, they will engulf it and break it down and process the antigens and display them on their surface. So using the MHC molecules, this antigen presentation serves as a signal to the adaptive immune system and alerts T cells and B cells that a highly specific foreign threat has been detected. So now I'm going to talk about clonal selection and expansion. So clonal selection is what gives the adaptive immune system its precision or specificity, and it begins when a V or T lymphocyte encounters an antigen that perfectly matches its unique receptor. So out of millions of potential lymphocyte clones, only one with the right receptor shape will bind to the antigen. And that single binding event activates the selected cell and triggers clonal expansion. So this is where the cell will rapidly divide and produce a large population of identical or clones cells. And these will differentiate into effector cells, which will fight the infection, and memory cells, which will remain in the body and for a long time and become stronger over time with exposure. So clonal selection and expansion are central to the adaptive immune response, and they're initiated by signals from the innate immune system. So like antigen-presenting cells and dendritic cells, macrophages, these are key players in the innate defense and process and present antigens to naive T cells, effectively choosing which clone will be activated. So cytokines produced by the innate cells also help shape how clones differentiate and determine whether immune response will lean toward antibody production or cell-mediated defense. So now I'm going to talk a little bit more about the antigen presentation and MHC cells. So MHC molecules act as the communication bridge between the innate and adaptive branches of the immune system. So when antigen-presenting cells, like macrophages, these engulf a pathogen, they process it and display it pieces of that pathogen called antigens on their surface using MHC class II molecules, and these displayed antigens are then recognized by helper T cells of the adaptive immune system, which will coordinate the next stage of defense. So then MHC class I molecules, these on the other hand, are used nearly by all body cells to present internal antigens. These include like the viral proteins or the cytotoxic T cells. This alerts the adaptive immune system to destroy infected cells and prevent further spread, so that MHC molecules can serve as the link between early detection and signaling roles of the innate immunity and highly specific and targeted adaptive immunity. So now I'm going to talk about adaptive immunity and vaccination. So adaptive immunity, I'm going to touch on this more in the second question, but adaptive immunity occurs when your body mounts its own defense. So this is either through infection or vaccination. And passive immunity, on the other hand, this provides short-term protection by transferring pre-made antibodies from another source. So vaccines are the main tool for artificial active immunity, and they train your immune system by exposing it to antigenic material. For instance, mRNA vaccines use lipid-encased genetic material that will produce a harmless antigen. So now, to round it up, I'm just going to talk about the comparisons between the innate and adaptive immune system. So the innate system is your body's first responder. It reacts quickly, generally using physical barriers like inflammation or phagocytosis. And the adaptive system, on the other hand, is slower and responds initially but highly specific. And it relies on B cells and T cells that recognize unique antigens and forms memory cells for lasting immunity. So what's important is that these two branches of the immune system don't work separately. They're tightly connected, always communicating with each other. Innate defenses will detect pathogens and signal adaptive mechanisms to respond more precisely and build immunity over time and strength and efficiency. So vaccinations take advantage of this by priming adaptive immunity and allowing for faster and stronger responses upon a real exposure. So these are my sources. Thank you so much.